is Laney Druhan. I'm a junior majoring in cell and molecular biology in Tulane School of Science and Engineering. I do synthetic biology research, which means I get to create DNA that has been acquired from multiple DNA sources. So I'm studying the potential to use CRISPR systems to temporarily silence genes. So you take like DNA from a bacteria and you can cut out the specific genes that you want and then insert a more favorable gene. Hi everybody, welcome to my lab. Let me show you around a few of our instruments. So all of my research deals with DNA, which is microscopic. So all of our pipetting and samples are pretty small. So we use these micropipettes. I'm just gonna pipette a little bit of media to show you. So as you can see, it's like very small amount. Yes, it's like specifically in genetic disorders that have like single mutations. So like sickle cell anemia is a really good example of this. It's like just one mutation causes such a drastic effect on the human. So you can use CRISPR systems to edit that one base pair mutation and fix the genetic disorder. And then if we want to visualize our DNA, I use gel electrophoresis for that. So it uses an electric current to move the DNA towards the positive end, and it can separate the DNA by size. So this is a gel that I made, and I can use one of the micropipettes to just pipette directly into the gel. As you can see, it's just kind of sitting on the gel there. And then I can just like adjust the voltage level and turn on the gel. And over time, about 30 minutes from now, it'll move down the gel, and then you could visualize it on a UV light and see what size your DNA is. When I took like my first genetics courses and I could see like how such small changes can lead to huge effects, I was like really interested in that. And so I knew I wanted to do some kind of like molecular biology research. And I heard about Dr. Sandoval's lab, which does like CRISPR gene editing, and I thought that that was really interesting and aligned with my goals and interests. So after we look at the gel on the UV light and we've confirmed that we have the DNA that we want, we can transform it into bacteria cells, and we can use their replication machinery to grow our DNA up into a higher quantity. And so once we have our bacteria cells, we can plate them onto these agar plates where they're able to grow up into an amount that we can actually see with our eye. And you just spread the bacteria culture out. We want it to be in a thin layer so that we can see like distinct colonies for the bacteria. And then you have to tape the plate shut so that it doesn't dry out or get contaminated by the air. And then I place it into this incubator overnight, which is at the temperature that bacteria like to grow at. Doing research has been really great for me. I think it has pushed me in ways that my classroom courses can't push me. Like I have to think of things in a more analytical way than I do on like an exam. I really like to know like the why and the how behind things, and I don't like to just take things at face value. And so research has allowed me to like answer those deeper questions that you can't just get from a lecture.